रही है। अरे ट्रेन को रोको, मुझे भी ट्रेन पर बैठा लो, नहीं तो एक खतरनाक बिल्ली मुझे काट लेगी। बापरे, लगता है ट्रेन छूट जाएगी और तेज दौड़ती हूँ। मुझे बहुत डराया है इस बिलैया ने। कमीनी बिल्ली, आजा आजा, अब आके काट ले मुझे। अरे वाह वाह पापा, ये दोनों आइसक्रीम तो बहुत मीठे हैं। बहुत मजा आ रहा है खाने में इन दोनों को। अरे चिंकी, तुम अकेले ही खा जाओगे क्या इन दोनों आइसक्रीम को? चिंटुआ और बाबू के लिए भी तो छोड़ दो। अरे बाबू पता नहीं कहाँ गया है, और चिंटुआ के ऊपर अलमारी गिर गया है। वो छत के ऊपर अलमारी के नीचे दबा हुआ है। क्या चिंटुआ ऊपर अलमारी के नीचे दबा हुआ है? अरे तो तूने पहले क्यों नहीं बताया मुझे? मैं आ रहा हूँ चिंटुआ तुम्हें बचाने के लिए। जब तक पापा चिंटुआ को बचा रहे हैं, तब तक इन दोनों आइसक्रीम को खा जाती हूँ। अरे चिंटुआ, तू कैसे अलमारी के नीचे दब गया? ये मत पूछो कि कैसे दबा, बस मुझे जल्दी निकालो यहाँ से। बहुत देर से यहाँ दबा हुआ हूँ। हाँ रुको, अभी निकालता हूँ। रुको पापा, क्या हुआ, क्यों रुका दिया मुझे? अरे पापा, तुम तो आइसक्रीम लेकर आए थे, तो जल्दी दो आइसक्रीम मुझे। अरे वो दोनों आइसक्रीम को चिंकिया ने ले लिया और वो उसे नीचे खा रही है। क्या? अरे तो जल्दी अलमारी को उठाओ ना, मुझे चिंकिया से आइसक्रीम छीनना होगा, नहीं तो पूरा का पूरा आइसक्रीम खा जाएगी। ठीक है, उठाता हूँ। अरे अलमारी तो बहुत ही ज्यादा भारी है, उठ ही नहीं पा रहा। अरे इतना देर हो गया, अभी भी ये बिल्ली मेरा पीछा कर रही है। अरे लगता है ट्रेन रुकने वाली है। अब क्या होगा मेरा? ये बिल्ली तो मुझे काट लेगी और उसके बाद 4-4 इंजेक्शन लगवाना पड़ेगा। लगता है ट्रेन पर से भी भागना पड़ेगा। भागो! अरे वो तो बाबू है। बाबू बाबू, जल्दी बुलडोजर को रोको, मैं भी आ रही हूँ। अरे वो तो मम्मी है, और वो बड़ी बिल्ली मम्मी को दौड़ा रही है। जल्दी आओ मम्मी, जल्दी आओ। आ गई, रुके रहना। अब जल्दी बुलडोजर को चला बाबू। हाँ चलाता हूँ। अरे मम्मी, तुम्हारे पीछे वो बड़ी बिल्ली क्यों पड़ी हुई है? अरे मैंने उसे डंडे से मारने की धमकी दी, तब से उसने मुझे बहुत दौड़ाया है। अरे बाबू, तो बुलडोजर को क्यों रोकने लगा? अरे मम्मी, मैं नहीं रोक रहा हूँ इस बुलडोजर को, ये अपने आप ही रुक रहा है। क्या? अरे तो जल्दी कुछ करना, वो खतरनाक बिल्ली पीछे ही है। लगता है बुलडोजर का तेल खत्म हो गया। क्या? कितना मजा आ रहा है गर्मी में ठंडा ठंडा आइसक्रीम खाने में। अरे पापा, क्या कर रहे हो? तुम लगा के उठाओ ना अलमारी को, नहीं तो चिंकिया नीचे वो पूरा आइसक्रीम खा जाएगी। अरे पूरा तो दम लगा रहा हूँ, लेकिन उठा नहीं पा रहा हूँ इसे। तुम बहुत ही कमजोर हो पापा, तुमसे एक अलमारी भी नहीं उठ पा रहा। तूने मुझे कमजोर बोला अपने पापा को, अब देख तू। या! उठ गया अलमारी, अब जल्दी निकल वहाँ से। चिंकिया, पूरा आइसक्रीम मत खाना, मैं भी आ रहा हूँ। मम्मी, अब हम चारों तरफ से घिर गए हैं। आगे पीछे दोनों तरफ बिल्लियाँ ही बिल्लियाँ हैं। वो छोटी बिल्ली तो और भी ज्यादा खतरनाक है। तुझे बोली थी कि बिल्लियों का घर मत तोड़ो। अब देखो ये दोनों बिल्लियाँ कितनी गुस्से में हैं। मम्मी, अब तो हम दोनों गए।
Excretion. Oligochaetus use miranifiria for excretion and for iron and water. regulation. As with polychaetus, funnels of metanephridia are associated with the segments just anterior to the segment containing the tubule and the nephridiopore. Nitrogenous waste include ammonia and urea. Oligochaetus excrete copious amount of very dilute urine, although they retain vital ions, which is important for organisms living in environments where water is plentiful, but essential ions are limited. Oligochaetes possesses collagen tissue that surrounds the dorsal blood vessel and lies over the dorsal surface of the intestine. Collagen tissue acts similarly to the vertebrate liver. It is a site of amino acid metabolism. Collagen tissues deaminates amino acids and converts ammonia to urea. It also converts excess carbohydrates into energy storage molecule of glycogen and fat. Reproduction and development. All oligochaetes are monoecious and exchange sperm during copulation. One or two pairs of testes and one pair of ovaries are located on the anterior septum of certain anterior segments. Both the sperm ducts and the oviducts have ciliated funnels at their proximal ends to draw gametes into their respective tubes. Testes are closely associated with three pairs of seminal vesicles, which are sites for maturation and storage of sperm prior to their release. Seminal receptacles receive sperm during copulation. A pair of very small ovisacs associated with oviduct are sites for the maturation and storage of egg prior to egg release. During copulation of lumbearers, two worms line up facing in opposite direction with the ventral surfaces of their anterior ends in contact with each other. The orientation lines up the clitellum of one mode with the genital segments of the other. Worms are mucus sheet that the clitellum secretes envelops the anterior halves of both worms and holds the worms in place. Some species also have penile structures and genital city that help maintain contact between worms. In lumbricus, the sperm duct release sperm which travel along the external ventral body walls in sperm grooves formed when special muscular contraction along the groove help propel sperm towards the opening of the seminal receptacle. In other oligochaetes, copulation results in the alignment of sperm duct and seminal receptacle opening and sperm transfer in direct copulation. Last 2 to 3 hours. During which both worms give and receive sperm. Following copulation, the clitellum forms a cocoon for the deposition of eggs and sperm. The cocoon consists of mucoid and chitinous material that encircle the clitellum. The clitellum secretes a food reserve albumin into the cocoon, and the worm begin to back out of the cocoons. Eggs are deposited in the cocoons as the cocoon passes the opening to the oviduct, and sperms are released as the cocoon passes the opening to the seminal receptacles. Fertilization occurs in the cocoon and the worm continues backing out. The ends of the cocoon are sealed and the cocoon is deposited in the moist soil. Skin of sensors of heat and cold. Sensors of temperature, thermoreceptors are also aware sensory nerve ending. Thermoreceptors may be present in either the epidermis or dermis. Mammals have a distinctly different distribution of areas sensitive to either cold or warm. These areas are called cold or warm spots. A spot reflects to a small area of the skin that, when stimulated, yield a temperature sensation of warmth or cold. Cold receptors in the skin respond to temperature below skin temperature. Material coming into contact with the skin need not be warm or cold to produce temperature sensations. For example, when metal is placed on the skin, it absorbs heat and you feel a sense of coldness. Wood placed on the skin absorbs less heat and therefore feels warmer than metal. The ability to detect changes in temperature has become well developed in a number of animals. For example, rattlesnakes and other pit vipers have heat-sensitive pit organs on either side of the face between the eye and nostril. These depressions are lined with sensory epithelium containing receptor cells that respond to temperatures. Different from the snakes, surrounding snakes use their pit organs to locate warm-blooded prey. Many animals rely on the tactile stimuli to obtain information about their environment. Mechanical sensory receptors in vertebrate skin detect stimuli that the brain interprets as light, touch pressure, and vibrations. Light touch is perceived when the skin is touched but not strongly deformed. Receptors of light touch include bare sensory nerve ending and tactile corpuscles. Bare sensory nerve endings are the most widely distributed receptors in the vertebrate body and are involved in the pain and thermal stimuli as well as light touch. The bulbs of crayons are mechanoreceptors found in the dermis in certain parts of the body. They respond to some physical stimuli such as position changes. Other receptors for touch, pressure as Pacinian corpuscles and the organs of Ruffini. Many animals have specially adapted sensory hairs called vibrissae on their wrists, snouts, eyebrows. Around the base of each vibrissae is a blood sinus nerves that border the sinus, carrying pulse from several kinds of mechanoreceptor to the bear of interpretation. The sense of smell or olfaction is due to olfactory neurons in the roof of the vertebrate nasal cavity. These cells, which are specialized endings of the fibers that make up the olfactory nerve, lie among supporting epithelial cells. They are densely packed. For example, a dog has up to 40 million olfactory receptor cells per square centimeter. Each olfactory cell ends in a tuft of cilia containing receptor sites for various chemicals. Several theories have been proposed to explain how odors are perceived. The most likely one is that odor molecules physically interact with protein receptors on the receptor plasma membrane. Such as interaction somehow alters membrane permeability and leads to a generated potential in most species. Opening in the snouts leads in the olfactory receptors. Recent research has revealed that some species rely heavily on their sense of smell. For example, salmon and lampreys return to swamp in the some streams in which they hatch yearly. Earlier, their migrations to these streams often involved distances of hundreds of miles. and are guided by the fish's perception of characteristics odor of their spawning stream. Olfaction is an important sense for many amphibians. It is used in mate recognition as well as in detecting noxious chemicals and locating food. Olfactory senses are better developed in the reptile